Hi, my name is Taylor Roberson. I'm a senior at WFS High School, and my lab research project was on establishing a relationship between productions of the neuropeptide F gene and addiction. So, some background information before we actually get to talking about my project. The neuropeptide F gene, which is what I'm focusing on this project, controls the decisions related to food in drosophila, which, for example, in humans, when we are hungry, we decide which food we want by what we like and prefer. So it's like that in drosophila. A group of scientists did a project related to this. They had two groups. One group that were allowed to have sexual activity with females, and one group that got rejected by the females. The whole point of this was to see if the sexually rejected drosophila would turn more towards alcohol than if they weren't sexually rejected. When they analyzed their results, they realized that if the drosophila were sexually rejected, they preferred the food with ethanol more than the regular food, where it was the opposite with the drosophila who actually had sexual intercourse. This is a chart of the life growth of drosophila. So they start off as eggs, progress to the larva, and then progress to pupa. After which, you can then easily identify which gender they're going to be. As you can see, an adult female is a little bit tan at the bottom, whereas on adult males, they're a little bit more darker. And the lifespan of the drosophila is around 2 weeks. My hypothesis for this lab research project is the production of the neuropeptide F gene will be higher in the case of sexual activity than in the other groups. My groups are isolated male drosophila with alcohol food and isolated female drosophila with alcohol food. Alcohol in these food groups were about 15 milliliters per 100 milliliters of food. Isolated male drosophila caffeine and isolated female drosophila caffeine with caffeine. The caffeine was about 0.135 grams for 100 milliliters of fly food. You also have isolated male drosophila with regular food and isolated female drosophila with regular food, just to see if maybe they get some neuropeptide F production from just plain old food, because humans get addicted to food, why not animals? Then there's also a combined group gender of drosophila, and they also have regular food. This is to see what the neuropeptide F reproduction is when there is sexual activity. My procedures were to let these groups live in their environment for a week so they can get the maximum amount of exposure to the environment they are in. Then after that week, you take them out of their vials, and using a mortar and pestle, you squish them and put them into an epi, and then you extract their RNA from them. And then after you extract the RNA, you run the RNA with a template DNA on the real-time PCR machine. So my results for this project have not yet been completed, though they will be fully analyzed and ready for you guys before Bio Expo. So in conclusion, the purpose was to analyze the neuropeptide F gene, which this project will do in the case of addiction. Because addiction is a big problem in today's society, so if we can figure out if it's actually a gene doing it, that could help us cure addiction faster. And results, again, will be fully analyzed before Bio Expo, so you guys can come and find out more. So I would like to thank Mr. Henry Weeks, who is my personal teacher at WFS High School in Advanced Molecular Genetics. And I would like to thank Miss Wendy Neal. She is also another teacher at Advanced Molecular at WFS High School, and she also teaches Advanced Molecular Genetics as well as does some research. I would also like to thank Mr. Mike Stratton, who teaches at Centralia High School, and he helped me fine-tune my essays and stuff for you guys. And I would like to thank STEM and the Shayless Foundation for funding this project. And I'd like to thank you guys for sitting and listening to this video and for judging us and helping us become better scientists. Can't wait to see you guys at Bio Expo. Thanks.